Hi, I'm Josh W., and today we're going to be talking about the protein folding process. If you've ever had any problems with it in class or a teacher just not understanding it or explaining it well enough, today we're going to get into a deeper understanding of it. So the starting point. As we know, proteins are built of amino acids, the main 20. They're also broken down and refolded constantly. This process of being broken down and refolded is called denaturation. This denatured protein is the protein that we'll be starting from, or the starting point. The first general step in protein folding is the hydrophobic collapse. This is the first step needed to get a denatured protein to a folded protein. This is the coming together of hydrophobic amino acids from far apart in the sequence. See this GIF? You see as they come together and close up? Now, weak bonds, no, not James Bond, but weak bonds are what hold the structures together. And there are three main types of weak bonds. There are electrostatic interactions, or ionic bonds or salt bridges, hydrogen bonds, which occur when hydrogen is covalently bonded to an electronegative atom, and van der Waals interactions, which happen with molecules with no charge nor polarity. And there are also disulfide bonds, which those are just a type of covalent bond that occur between two sulfur atoms. Now, interactions can either be long range or locally. Long and local range interactions can take place at the same time, and this causes the formation of the native state. Remember the native state, this is important. Now, as we discussed earlier, the hydrophobic effect draws together hydrophobic amino acids, and this is the long range or tertiary structure. Local interactions pertain to the secondary structure formation through hydrogen bonding or close together. Now let's take a deeper dive into the structures formed. So proteins can be categorized into four main groups. We have our primary structures, our secondary structures, our tertiary structures, and our quaternary structures. These structures are formed due to different actions and interactions during folding and subsequently the unfolding and repetition of this process. Here's a better picture. Right here, we have our primary structure, our secondary structures, our tertiary structure, and our quaternary structure, which is multiple structures combined into one. And not all folds are folded equally. No, but seriously, some folds are actually incorrect and thus must be unfolded and tried again. There are chaperone proteins that exist and they're able to watch over this and protect the hydrophobic residues. Intermediates also have a huge role in defining how a protein folds. Now they're always looking, and when we say always looking, we mean that while the structures are folded, unfolded, and refolding, they're constantly looking for the lowest energy state or the most stable structure. Now this is known as the native state. Remember, we said earlier that the native state is important, and this is what becomes a functional protein. Right here, we have our native state, and this is the denatured state. Here, as you can see, we're looking for our lowest energy level. We're always looking, okay? The native state's always looking. And clearly you can see our pissed off denatured state. All right, well, I hope you enjoyed the video and I hope your understanding of protein folding process has gotten better.